Hello everyone, welcome to the session on the metabolism of ketone bodies. Yes, so what are ketone bodies? Ketone bodies are alternate source of fuel for certain tissues like brain, skeletal muscle and cardiac muscle. We all know glucose is the main source of energy for all the cells. Apart from glucose, ketone body is the next source of energy for certain cells like brain, okay, skeletal muscle, cardiac muscle and the renal cortex. If you see brain cells, so preferably brain will use only glucose as its source for energy. If that glucose is not available, the brain cells will utilize ketone bodies as the source of energy. And there are certain cells which depends only upon glucose for energy. For example, red blood cells, because that does not have mitochondria, it depends only on glucose, especially the glycolysis pathway for energy. Okay, and lens, cornea, these are certain cells which solely depends upon only glucose for their energy. So these ketone bodies are alternate sources of fuel for certain tissues. Yes, so we will see what are those ketone bodies. The three ketone bodies are acetone, acetoacetate, beta hydroxybutyrate. So these are the three ketone bodies. Actually, the ketone bodies itself a misnomer. Okay, yes. So if you see which is the functional ketone body among these three, acetoacetate and beta hydroxybutyrate. So these two are the functional ketone bodies. Acetone being highly volatile, it gets exhaled via lungs. And these two, acetoacetate and beta hydroxybutyrate, they are organic acid and they are water soluble. And the primary ketone body being the acetoacetate. Beta hydroxybutyrate is derived from acetoacetate. So these two are the functional ketone bodies. The question can be, which among the following is a functional ketone body? Okay, the option can be acetyl-CoA, uh, palmitic acid, okay, retinyl palmitate and acetoacetate. So the question can be asked this way. All of the following are ketone bodies except acetone, acetoacetate, beta hydroxybutyrate, acetyl-CoA. Okay, so acetyl-CoA is the wrong answer. So these three ketone bodies you have to remember. Yes. So when these ketone bodies will be produced in the body, okay, there are certain situations which favors ketogenesis, that is ketone body synthesis. If you see here, we saw the oxidation of fatty acid in the previous session where the 8-acetyl-CoA which is produced at the end of oxidation of palmitic acid will enter into TCA cycle for further complete oxidation, right? Yes, if suppose that is not happening, okay, if the intermediates of the TCA cycle are unavailable, the acetyl-CoA which is formed at the end of fatty acid oxidation will be shunted to ketone body synthesis. Okay, the acetyl-CoA formed in the liver, okay, this ketogenesis happens in the liver. The acetyl-CoA formed in the liver during the oxidation of fatty acid can either enter into TCA cycle for complete oxidation or ketone body synthesis. Okay. Yes. So, if the acetyl-CoA cannot enter TCA cycle, the acetyl-CoA which is generated by the oxidation of fatty acid will enter into ketone body synthesis. Okay. So, they are produced in the liver when the amount of acetyl-CoA exceeds the capacity of the TCA cycle to oxidize it completely. Okay. Yes, we will see the steps of ketone body synthesis from acetyl-CoA. So first what happens, two molecules of acetyl-CoA will combine and form acetoacetyl-CoA. If you see here, this is one acetyl-CoA, there is one more acetyl-CoA. Okay, both the acetyl-CoA will combine, will form acetoacetyl-CoA with the help of enzyme thiolase. And this ketone body synthesis is happening inside the mitochondria. Because we know mitochondria is the site where fatty acid oxidation is occurring. Similarly, ketone body synthesis also occurs inside the mitochondria because the fatty acid which is getting oxidized with the help of beta oxidation enzymes will get converted to acetyl-CoA. TCA cycle enzymes are also present in the mitochondria. If the cell requires energy, that will enter into TCA cycle and generate energy. If it cannot enter TCA cycle, it goes through ketone body synthesis within the same mitochondria to generate ketone bodies. So the ketone body synthesis is happening inside the mitochondria. Okay. Next, this acetyl-CoA will accept one more or will react with one more acetyl-CoA. Okay, if you see here, this acetoacetyl-CoA will react with one more acetyl-CoA and it forms HMG-CoA, hydroxymethylglutaryl-CoA. The enzyme here is very simple, HMG-CoA synthase. Okay, HMG-CoA synthase which generates hydroxymethylglutaryl-CoA. Okay, yes. So far we have synthesized. If it cannot enter TCA cycle, the acetyl-CoA, okay, this acetyl-CoA will two acetyl-CoA combine and form acetoacetyl-CoA with the help of enzyme thiolase. Next enzyme HMG-CoA synthase will accept one more acetyl-CoA and it forms hydroxymethylglutaryl-CoA. The next enzyme hydroxymethylglutaryl-CoA lyase. Okay, HMG-CoA lyase will convert this HMG-CoA to, okay, acetyl-CoA and acetoacetate. Okay, one acetyl-CoA is released. The enzyme here is HMG-CoA lyase, and this acetoacetate will be converted to beta-hydroxybutyrate with the help of enzyme beta-hydroxybutyrate dehydrogenase. As the name indicates, the reducing equivalent NADH will be utilized in this reaction, and this reaction is a kind of reversible reaction. If you see here, yes, it is a reversible reaction. Acetoacetate will be converted to beta-hydroxybutyrate by consuming one NADH. Okay, the enzyme here is beta-hydroxybutyrate. So, beta-hydroxybutyrate is the end product or is the final product formed in ketone body synthesis. Okay, all the reactions are happening inside the mitochondria. We saw the same enzyme, right? HMG-CoA synthase, the same enzyme, the same set of reactions which is happening in the cytosol for cholesterol synthesis, right? Yes. Acetyl-CoA will combine and form acetoacetyl-CoA. Acetoacetyl-CoA will take one more acetyl-CoA and it forms HMG-CoA. Till this step, it is kind of similar between cholesterol synthesis and ketone body synthesis, but the difference is cholesterol synthesis is happening in the cytosol, whereas ketone body synthesis is happening in the mitochondria. Okay, yes. So far we have synthesized ketone bodies. Okay, so the end product of ketone body synthesis, that is either acetoacetate or beta-hydroxybutyrate, is the end product of ketogenesis. Which one will be the end product is determined by a ratio called NADH NAD+. So when there is more NADH, beta-hydroxybutyrate is the end product of ketone body synthesis. Okay, when there is more NADH, beta-hydroxybutyrate is the end product. When there is less NADH, acetoacetate is the end product, is the final product of ketogenesis. This you have to remember, because in the previous year one of the MCQ is all of the following statements are true regarding ketone body synthesis except the options were related to NADH NAD plus ratio. Okay, so for this you have to know this NADH NAD plus ratio and whether it acetoacetate or beta hydroxybutyrate is the final product of ketogenesis. If you see here, if there is more NADH, beta hydroxybutyrate is the end product. If there is less NADH, acetoacetate is the end product of ketogenesis. This statement have to be clear. Okay, since when the fatty acids are getting oxidized, we know one NADH and one FADH2 is generated. So there will be more NADH when the fatty acids are getting oxidized. Okay, so in that case, the final product which is formed via ketogenesis is beta hydroxybutyrate. Okay, so beta hydroxybutyrate is the final product. Okay, because this ratio, okay, here I have mentioned NAD plus NADH ratio, don't get confused. This one will be quite easy for you to remember. Since this NADH NAD plus ratio will be more when the fatty acids are getting oxidized, the final ketone body which is formed will be beta hydroxybutyrate. So the normal blood level will be less than 3 mg per deciliter and the unit will be less than 125 mg per deciliter. So what is the fate of this ketone body? This ketone bodies which is getting generated in the liver will move on to the extrahepatic tissues like skeletal muscle, cardiac muscle, brain, renal cortex, where these are the alternate source of fuel for those tissues. So this ketogenesis which is happening in the liver will transfer or will transport the ketone bodies which is formed in the liver to the tissues where required. For example, we keep that as muscle, skeletal muscle or cardiac muscle. So what is the fate of those ketone bodies? This is called ketolysis, okay, or ketogenolysis, that is breakdown of ketone bodies. In the liver, ketone bodies are getting synthesized, so that is ketogenesis. In the extrahepatic tissues, it is getting utilized. It is ketone body utilization or ketogenolysis or breakdown. Okay, so we will see how it is getting broken down in the extrahepatic tissues. Suppose if it is getting transported as beta hydroxybutyrate, it has to be converted to acetoacetate again. Yes, so in that scenario, what happens is one NADH which is utilized in the liver will be regenerated here, right? Yes. So 3-beta hydroxybutyrate will be converted to acetoacetate with the help of enzyme beta hydroxybutyrate dehydrogenase. Okay, this acetoacetate, it is just acetoacetic acid. So it has to be activated or a coenzyme A should be attached to this. That will be done by an enzyme called CoA transferase. Okay, acetoacetate succinyl CoA transferase. If you see here, from succinyl CoA, the coenzyme A is getting transferred to acetoacetate to form acetoacetyl CoA. So the enzyme is CoA transferase. So this enzyme will transfer coenzyme A from succinyl CoA to acetoacetate, which will result in the formation of succinate and acetoacetyl CoA. And this acetoacetyl CoA will be broken down by the same enzyme thiolase to form two molecules of acetyl CoA, and this acetyl CoA will enter into the TCA cycle in the extrahepatic tissues, will generate 20 ATP. So that ATP will be utilized for those will be utilized by those extrahepatic tissues. So this is how ketone bodies which is produced in the liver is transported to the extrahepatic tissues, and it is broken down to two molecules of acetyl CoA, and those two molecules of acetyl CoA will enter into TCA cycle, and it will provide energy to that tissue. So what is the significance? So why the liver, even though it produces ketone body, it cannot utilize ketone body? Okay, the question is, even though the liver can generate ketone body, it cannot utilize ketone body because it lacks a specific enzyme, the main enzyme which helps in activating acetoacetate. Okay, we saw CoA transferase, right? Yes, so that is called, otherwise called thiophorase. The other name of that enzyme, acetoacetate succinyl CoA transferase enzyme, is called thiophorase. Okay, this thiophorase, this is deficient in liver. That's why liver cannot utilize ketone body. Even though it can synthesize ketone body, it cannot utilize ketone body because it lacks an enzyme called thiophorase. Okay, so this is the enzyme thiophorase, the one which transfers the CoA group. Okay, yes. So this is a very important MCQ segment. Why a liver cannot utilize ketone body. Moving on to the regulation part. The regulation happens at 3 levels. Anything which affects the generation of free fatty acid is going to affect the generation of ketone body. So if you see here, the regulation happens at 3 levels. The first one is lipolysis, which is occurring in the adipose tissue, because adipose tissue is the source of free fatty acids which is coming to the liver. Okay, the factors which are going to affect the lipolysis in the adipose tissue. The second one is, we know malonyl CoA is the gatekeeper of carnitine acyl transferase 1. So when there is more malonyl CoA, carnitine acyl transferase 1 will be inhibited. Okay, so when there is more lipolysis, fatty acid synthesis will not occur. More free fatty acids will be available in the liver. Okay, this gatekeeping work will not be done by malonyl CoA. Okay, this is the second level of regulation. Okay, third level is whether the acetyl CoA should go to ketone body synthesis or it should go for TCA cycle. Okay, that is third level of regulation. Yes. Yes, so free fatty acids are the precursors of ketone bodies because those free fatty acids are oxidized in the mitochondria, converted to acetyl CoA, okay, and that acetyl CoA either it go for TCA cycle or it goes for ketone body synthesis. So anything which regulates lipolysis or generation of free fatty acids will also regulate ketone body synthesis. Okay, the main thing is the ratio. Okay, insulin glucagon ratio. We know insulin favors lipogenesis, whereas glucagon favors lipolysis. So whenever there is more glucagon to insulin ratio, there will be more lipolysis. So more free fatty acids will be mobilized from adipose tissue to the liver for oxidation, and that will be converted to ketone bodies eventually. So more lipolysis, more free fatty acids in the plasma, that goes to the liver, finally it generates ketone bodies. Okay, yes. So insulin will favor lipogenesis, glucagon will favor lipolysis. How insulin favors lipogenesis? It allows the cell to take up glucose and it activates acetyl-CoA carboxylase so that fatty acid synthesis occurs. Okay, yes. Glucagon will inhibit acetyl-CoA carboxylase and also it enhances lipolysis, so there will be more free fatty acids available in the cell. So those free fatty acids will go in for oxidation because synthesis will not occur, so no malonyl CoA. So the gatekeeper work of malonyl CoA will not be done. Okay, so it results in more oxidation of fatty acid. If you see clearly here, the free fatty acids from the blood are coming to the liver. When there are more free fatty acids, these are the end product of actual synthesis, right? Yes. So this acyl-CoA will inhibit the acetyl-CoA carboxylase. So acetyl-CoA carboxylase is inhibited, so no malonyl-CoA is produced. Since there is no malonyl-CoA, the carnitine acyl transferase 1 is not inhibited, it is open. So all the fatty acids will enter via carnitine acyl transferase 1 via CPT1 into mitochondria, and these fatty acids are undergoing oxidation. Okay, beta oxidation, and it gets converted to acetyl-CoA. It should either go to TCA cycle or ketone body synthesis. Okay, this is how insulin and glucagon plays a role in ketone bodies regulation. So ketosis means when there is excess of ketone body in the blood and that is getting excreted in the urine. So it is called ketosis. Ketonemia is excess of ketone body in the blood. Ketonuria is excretion of those ketone bodies in the urine. And the person will also have smell of acetone in the blood because this is the one which is highly volatile. Okay, this is the one highly volatile, and the acetone will be produced from acetoacetate spontaneously without any enzyme by decarboxylation reaction, and that acetone is volatile. That will be exhaled via lungs. Okay, yes. So what are the causes of ketosis? Ketosis means ketonemia, ketonuria, and the smell of acetone in the breath. This is because of acetone, and which is formed spontaneously from acetoacetate by decarboxylation reaction. And causes of ketosis is one, starvation. Second one is uncontrolled diabetes mellitus. So these are the two causes of diabetic ketoacidosis, especially type 1 diabetes mellitus. Yes, so what happens is, since in starvation and type 1 in diabetes mellitus, there will be increased glucagon and insulin ratio. Yes, starvation, insulin will be less, glucagon will be more. So more glucagon and insulin ratio, more fat lipolysis in the adipose tissue. There will be free fatty acids so there will be synthesis of free fatty acids or production of free fatty acids in the adipose tissue, and those free fatty acids will come into the liver, and those free fatty acids will inhibit acetyl-CoA carboxylase. So malonyl-CoA will not be formed. The carnitine acyl transferase will be free. So it allows all the fatty acids into the mitochondria that will all undergo oxidation and produce acetyl-CoA. Okay, so there will be more and more NADH, right? So there will be increased NADH NAD plus ratio. If you remember in TCA cycle, the final step where malate is getting converted to oxaloacetate, in that reaction, one NADH is produced. Yes, so since we already have more amount of NADH, what happens? All the oxaloacetate will be converted to malate. Okay, all the oxaloacetate will be converted to malate. So there will be fall in the concentration of oxaloacetate, particularly within the mitochondria. So since there is no oxaloacetate formation, means since there is less amount of oxaloacetate in the mitochondria, instead of going in for TCA cycle, this acetyl-CoA will go in for ketone body synthesis. Okay, yes. So there will be increased NADH NAD plus ratio because of fatty acid oxidation. And this NADH NAD plus ratio has resulted in conversion of all the oxaloacetate to malate, and oxaloacetate is not available to start TCA cycle. So all the acetyl-CoA will go in for ketone body synthesis. Okay, yes. So, what are the clinical features of ketoacidosis? If you see, the one among the ketone body is acetone, which is highly volatile. So, the body tries to wash out all the acetone which is formed. So, the breathing will be very rapid and it will be very shallow. And this type of breathing is called Kussmaul breathing. Okay, Kussmaul's breathing. That is a typical acidotic breathing where the person will be breathing very fast and it is shallow. This is because ketone bodies are organic acids, right? Yes, so ketone bodies are organic acids like acetoacetate, acetic acid, beta hydroxybutyric acid. So accumulation of acid will result in metabolic acidosis. Okay, the pH will drop down, and this drop in pH is because of excessive accumulation of acid. So in order to compensate that, there will be respiratory alkalosis, and that is rapid and shallow breathing, that is Kussmaul's breathing. So the compensatory hyperventilation is to clear, it is because of the metabolic acidosis which has set in already due to the accumulated ketone body. And the person will also have smell of acetone, that fruity odor. Okay, and urine also will contain acetone. Okay, urine in the diabetic ketoacidosis will also have fruity odor because of acetone. The person will also have nausea, vomiting, abdomen pain, increased thirst, confusion. These are all because the excess of the body tries to eliminate the excess of ketone bodies. So these ketone bodies are excreted in the form of sodium salt of in the form of sodium salts like sodium acetoacetate, sodium hydroxybutyrate. Okay, so there will be hyponatremia. This hyponatremia will result in the features of nausea, vomiting, increased thirst, okay, abdomen pain. So these are the clinical features of ketoacidosis. How will you diagnose? So we should look for the presence of ketone bodies in the urine. The test which is used for that is Rothera's test. This can be an MCQ. The test used to pick up ketone bodies in the urine is Rothera's test. Okay, the treatment for diabetic ketoacidosis is providing insulin and glucose. Yes, so that's all about the metabolism of ketone bodies. The MCQ segment here is name of the ketone bodies: acetone, acetoacetate, beta hydroxybutyrate. Why liver cannot utilize ketone body? Because it does not contain an enzyme called thiophorase. And regulation. And regulation, you have to read the regulation in the aspect of like reasoning out. So there is increased NADH and NAD plus ratio because of increased oxidation of fatty acids. So that will result in the shift of all the oxaloacetate to malate. So TCA cycle will not occur because oxaloacetate is not available. So everything is getting shunted to ketone bodies. So NADH and NAD plus ratio is quite important, and you can anticipate questions from that aspect. And finally, the clinical features of diabetic ketoacidosis. If you see in the last year, they have given a case scenario of diabetic ketoacidosis and they have asked for the diagnosis. Like which ketone body will be present in the urine and which test can be done to detect these ketone bodies. So acetone, it is highly volatile, it is exhaled via lungs, and the other two ketone bodies, acetoacetate and beta hydroxybutyrate, among these two, which is the predominant ketone bodies found in the urine, in the blood in case of starvation and diabetic, in the case of diabetic ketoacidosis, it is beta hydroxybutyrate. So these are the possible questions with respect to ketone body metabolism. Okay, thank you.